This is my video on translation. Translation is a process where the information from DNA stored in mRNA is converted into a polypeptide. The process of translation involves a type of RNA called tRNA and ribosomes. First, I will talk about the tRNA. The tRNA is said to be the link between the mRNA and polypeptide. The tRNA consists of a sequence of 3 nucleotides known as anticodon that are opposite and fit with codons from the mRNA. The tRNA also consists of an amino acid site which holds the amino acid produced from the codon on the mRNA. Ribosomes are also involved with translation. There are 2 types of ribosomes. So there's the large ribosomal unit and the small ribosomal unit. These ribosomes always exist in the cytoplasm as 2 pieces before coming together in the process of initiation. The large ribosomal subunit consists of 3 sites: the acceptor site, where new tRNA are attached, the polypeptide site, where the polypeptide is formed, and the exit site, where the tRNA are detached. The first process of translation is called initiation. First, the mRNA in the cytoplasm and the small ribosomal subunit attach together at the guanine cap at the 5' end of the mRNA. Next, the first tRNA with anticodons UAC and amino acid methylthionine attaches to the start codon AUG. This then allows the large ribosomal subunit to attach to the mRNA, and this is called the initiation complex. The second process of translation is called elongation. During elongation, the tRNA with its anticodon attaches to the A site on the large ribosomal subunit. The large ribosomal subunit then creates a polypeptide bond between the first amino acid in the P site and the second amino acid in the A site, creating a growing polypeptide. There we go, growing polypeptide. This then means that the first tRNA is no longer needed, so it detaches from the mRNA and its amino acid. This tRNA can now return to the amino acid pool to find another amino acid that matches its anticodons. After this, the entire ribosome translocates 1 codon on the mRNA from 5' to 3' direction. The growing polypeptide is now hanging off the last tRNA in the P site. This process keeps on repeating as the polypeptide is created. I will show one more process of this cycle. So these amino acids it will add on, creating our growing strain. So again, another amino acid will be attached to the open A site by a tRNA. A peptide bond will be formed between the next 2 amino acids and the polypeptide chain. And lastly, the tRNA in the P site detaches in the E site and is recycled. And the ribosome also translates 1 codon. Alright, so the last process in translation is called termination. So termination occurs in the ribosome once the ribosome encounters a stop codon, which can be UAA, UAG, or UGA. In this case, it is UAA. This stop codon does not code for any amino acids and does not have a tRNA which can attach its anticodons to it. This causes a protein release factor to attach to the stop codon at the A site and causes a water molecule to attach to the protein chain. So this allows the polypeptide to detach from the mRNA and the ribosome to separate from the mRNA. The mRNA and the ribosome are then recycled. So here is our polypeptide chain. So this was also gone, but now all of these are gone to be recycled. So we're left with our amino acid or polypeptide chain. So after the protein is released, it will go to the endoplasmic reticulum and finish synthesis there. The protein will fold into its 3D shape and may undergo other processes such as proteolysis, where the polypeptide is cleaved into fragments which form into different shapes. So the protein is then sent to the lysosomes by the Golgi apparatus and can end up being secreted from the cell. So this is the full process of translation. So we end with like a polypeptide chain, and then our ribosomes get recycled.